So we're going to talk about something a little different today, more cell biology maybe, um, and that is how cells are organized, complex cells are organized, and how molecules, particularly proteins, get to their destination or where they need to be. Um, so let's start by looking at a schematic of a cell here on the left, and we see all the different organelles or subcellular uh, organization. Of course, the nucleus where DNA is, and that's surrounded by a membrane that's contiguous with the endoplasmic reticulum, which is where secreted and membrane-bound proteins are synthesized, and we'll talk about that at length. And then the Golgi right above that, where proteins are sorted out so that they can be sent to their final destinations. We have lysosomes, which degrade molecules or incoming, in phagocytic cells, they degrade bacteria as well, or other invasive things. The mitochondria, where energy is made, or and a few other minor organelles. Now on the right is an electron micrograph of a cell, and we see some of these organelles. At the top, we see half of the nucleus. These dark spots are lysosomes. We see some mitochondria, as well as some other organelles. Okay, so, we used to think of cells as just a big bag of chemicals that floated around looking for the molecule that it needs to interact with, but we now really understand that this is a highly structured and very programmed machine, and so all these molecules are directed to where they need to be rather than letting them just float around and find their home accidentally. Now this is just an example of one type of cell. This is a liver, represents a liver hepatocyte, and showing how much the volume of the cell is taken up by the different organelles. And liver is a somewhat typical cell. It carries out a lot of different metabolic processes. So more than most cells. So in that way, it might be a little different, but similar to a lot of cells that aren't carrying out some kind of specialized function. So in the liver or hepatocyte, over half of the volume is in the cytosol. And it's not surprising. That's where a lot of these metabolic pathways are found. About a quarter of the volume is in the mitochondria. Then there's a small amount in the rough ER, an even smaller amount in the smooth ER. The nucleus takes up a fairly small fraction. The size of the nucleus is probably somewhat similar from cell to cell. So it's the size of the cell that determines what fraction of that is taken up by the nucleus. And then these other smaller organelles that are involved in metabolizing different compounds take up a very small fraction of the cell. Now this is a comparison of a hepatocyte, the one we just looked at, with a pancreatic cell, which is involved in synthesizing and secreting a lot of proteins that are important in secreting them into the digestive system where they're utilized. And so we see the biggest difference here. And now um, we're looking at the amount of membrane that goes into each of these organelles. And we see with the plasma membrane or cellular membrane, a little bit more in the pancreas, but the big difference is in the rough ER, and that's where secreted proteins are synthesized. So it's not surprising that there would be more ER, particularly rough ER, in a cell that was involved in secreting proteins. And I'll show you an example in just a second, actually on the next slide, where we can change the amount of rough ER in a cell based on how much protein it needs to accommodate. Okay, the smooth ER counts for a very small fraction of the pancreatic cell, especially compared to the hepatocyte. And again, there's a lot of lipid metabolism going on in the hepatocyte, and that's where lipid metabolism takes place. So it's not surprising that there's a relatively robust amount of smooth ER. Now, this is an electron micrograph of a cell that overproduces the enzyme HMG-CoA reductase. And of course, you all remember that that's the rate-limiting step in cholesterol synthesis, and again happening on the endoplasmic reticulum. And if you make a lot of extra HMG-CoA reductase, the cell will accommodate that by making more rough ER. There's another picture of a yeast cell that's overproducing the same enzyme, and you can see the ER surrounding the nucleus in the big picture, and then zoom in on this picture on the bottom of the layer after layer of rough ER to accommodate the extra HMG-CoA reductase. Now, when we look at a cell, we see that there are a number of compartments in the cell that are separated by lipid membranes. And so once a protein, or any molecule for that matter, crosses one of those membranes, it's topologically outside of the cell. And in order to get back into the cell, it would have to recross a membrane, which rarely happens. But what can happen is it can move from one chamber in the cell to another by budding off from one organelle and forming a vesicle, and then fusing with another membrane in another organelle. So in order to go from one organelle to another, protein or other molecule needs to cross a membrane just once, and then it can move to different compartments. And again, it would be topologically out of the cell, and it can be transported outside of the cell, again, without ever crossing another membrane, just by fusing that vesicle to the cell membrane. This is an important concept to understand how cells work. So when we talk about protein trafficking in the cell, this is kind of a roadmap of where proteins are going, and the different kinds of transport that we see. And we're going to talk about these in more detail, but basically there's three types of transport shown here up in the left-hand corner, shown with a red arrow, which is been labeled gated transport, is the transport of proteins in and out of the nucleus. And proteins don't need to cross a membrane in order to do that, but they do need to pass through gated ports in a membrane. And we'll talk about that in a little more detail. Now, proteins that are synthesized in the cytosol in order to get to either the mitochondria or the ER need to cross the membrane, and we'll talk about how that's done. They also move into the peroxisomes by crossing the membrane as well. I'm not going to talk about plastids because those are found in plants, and we're going to ignore plants for the time being. Now, once proteins have gone into the ER, then they can move around to different subcellular locations by vesicular transport and don't need to cross another membrane. So they can move into the Golgi where they are sorted out and sent out to the lysosomes or endosomes or secretory vesicles or straight to the cell surface. So if we have a vesicle shown at the top here with little purple proteins in it, that can bud off a smaller vesicle containing those proteins, which then can go and fuse to another membrane, releasing those proteins into another subcellular organelle. So a simple budding off and then fusing to another membrane can transport these molecules from one organelle to another. Now, of course, we need to have some signals to direct this, and there are a couple of signals that get things going. In other words, direct the protein to the membrane surface, either to the ER or to mitochondria, or many proteins have a signal at the very beginning, at the amino terminus, that directs them to the surface of a membrane. However, some proteins have an internal sequence that can be, as shown here, parts of it in different regions of the protein, and when the protein is folded, then they come together to form this signal. So if we look at the first type of signal, the amino terminus signal, we see there's a lot of ambiguity here. So there's no one run of amino acids that signals the protein to a certain location. Directing proteins to the nucleus might be a little more consistent, or a little more the same from protein to protein, shown here. But import into the mitochondria, we see a lot of differences, as well as import into the ER. So don't really want to sit down and memorize any of these things. Just be aware that these signals, these are signals on the front end of a protein. They vary quite a bit from protein to protein, but they do direct the protein to the appropriate membrane in order to pass through that membrane. Okay, I want to talk a little bit now about transport in and out of the nucleus. Again, this is a gated transport. It does not pass directly through a membrane, but rather through pores in the membrane. And if we look at the schematic of a nucleus, again, we see the nuclear membrane is contiguous with the endoplasmic reticulum, but it also has these pores in it, and they're not simply just holes in the membrane. They're a gated entry and exit point, which is shown in the next figure here, and on the right are some electron micrographs showing the pores, and on the left a schematic. And again, there are proteins that control the import and exit of different molecules, particularly big molecules. Small molecules pretty much can flow in and out through the pores. And that's shown in this figure. Again, small molecules can enter the nucleus by diffusion, but these large proteins have to be actively transported. And there are some proteins that facilitate that. This shows some schematics. So on the left, the cargo protein, that's the protein that needs to be moved into the nucleus, is associated with a receptor that binds that protein, and then that carries it into the nucleus. Often these receptor proteins are regulated so that under certain conditions they move proteins into the nucleus. Under other conditions they don't. And there's an example shown here. Actually, in this case, the protein being transported is modified during T cell activation. There's the nuclear factor AT. When it's phosphorylated, it is outside in the cytosol. Under high calcium concentrations, the phosphates are removed. That produces a transport signal. Then that allows that protein to move through the pore into the nucleus, and then it can bind to DNA and activate transcription. Now, if you lower calcium levels in the cell, then the protein gets phosphorylated, and then it is removed from the nucleus through a pore, again by an active process. So in this way, cells can regulate what in or out of the nucleus, and therefore regulate the transcription of different genes by sequestering these activating proteins outside of the nucleus in the cytosol when the cell doesn't want transcription of the target genes, and moving them into the nucleus in order to activate transcription. Okay, transport to the mitochondria. Then now these proteins have to cross a membrane. And as you may remember, the mitochondria is made up of two layers of lipid bilayers or membranes. So there's an inner membrane and an outer membrane. So there's space inside the inner membrane. There's a space between the two membranes, and then there's the outer membrane. And so we find proteins in all of these spaces, and also spanning some of the spaces. So we'll find proteins on the outer membrane, on the inner membrane, between the two membranes, inside the inner membrane, some that span both membranes. And so in order to put those proteins where they're needed, there are a lot of signals that go into this. Again, there's a signal at the amino terminus. Sorry if I make a mistake, call it the 5' end of it. You can see my orientation is more at the gene level than the protein level, but this is a typical signal sequence. And so in this case, when the alpha helix formed by this protein puts a number of positively charged amino acids on one side of the helix. So you can see that in the bottom in the coil here, shown as the red amino acids that all line up on the same side, with the yellow or nonpolar residues on the opposite side. Now there are a number of translocators in the mitochondrial membrane. These are called TOMs and TIMs. TOM for transport outer membrane and TIM for transport inner membrane. And these facilitate the transport of the protein through the membrane. So proteins can pass through both membranes at the same time, and so generally what happens here is when an mRNA is made for a mitochondrial protein, it moves into the cytoplasm. It's translated into a protein. That protein then has a signal sequence on it. It's recognized by a TOM, shown on the left of this figure. The blue and blue. It binds to that protein through the signal sequence and then passes it through the outer membrane. Now, if it needs to go into through the inner membrane, it will hook up with the TIM and pass the protein through at the same time. Once it's in the inner matrix, then that signal sequence is cleaved off and cleaved into amino acids, which can be recycled. So transport into the mitochondria requires energy, so it takes ATP to push the protein through the membrane. There's also a series of proteins that stabilize the nascent polypeptide chain in the cytoplasm. These are called HSP70 for heat shock protein 70. The heat shock proteins are a bunch of proteins that have some different functions, but many of them function as what are called chaperones, either to help the nascent protein fold into its native conformation or to stabilize the protein. And so they are induced when the cell is under some kind of stress, and heat being one form of stress. So these were first identified when cells were heated to a higher temperature, and they started to produce a whole series of proteins. And if you size them on an electrophoresis gel, HSP70 looks like it's about molecular weight of about 70,000, and so that's where the name came from. So once they're pushed through the mitochondrial membrane, and those are released. Again, this is a process that requires some energy to shove it through. After that, then the signal peptide at the front end is cleaved off, and the protein goes to its ultimate location. Okay, in order to move a protein through both membranes of the mitochondria, we require multiple signals. Again, the red on the left-hand side, the red at the amino terminus of the polypeptide signals for the protein to go through the membrane. And then there's a second signal, in this case directing it back to the inner membrane and anchoring it there. Also a signal on a protein synthesized in the mitochondria can direct it into the same conformation. The other possibility is to send the protein shown on the right here through the outer membrane and then anchor it with this orange sequence in the inner membrane, so that most of the protein is between the two membranes. So by having a number of different signals directing the protein to different locations, we can have it in either membrane, between the membranes, spanning both membranes, or in the matrix of the mitochondria. Okay, I'm not going to talk about much about proteins going in the peroxisomes. The mechanism is very similar to going into mitochondria. So once the protein enters into the peroxisome, new peroxisomes can be formed by increasing the size and volume of the peroxisome and then budding off into two daughter peroxisomes. Okay, now the bulk of proteins that are going to cross a membrane go through the endoplasmic reticulum into the Golgi and then are sorted out and sent to their final destinations. And this involves a little bit different mechanism. And when I was in graduate school, there was quite a big controversy about how proteins got through the membranes. And there were two major research groups, one at UCLA led by William Wickner and the other at Rockefeller University headed by Gunther Blobel. And so they were going back and forth about who was right about how proteins were sent through the membrane, and it turned out that they were both right because Wickner's group was studying transport across the mitochondrial membrane, and Blobel's group was studying the transport into the endoplasmic reticulum. So this is some pictures of the ER in a pancreatic cell that makes and secretes large amounts of digestive enzymes. And again, it's going to have a lot of rough ER because it's making a lot of protein that's ultimately going to be secreted out of the cell. So this is a basic mechanism of transport into the ER. And so the ribosome binds to a mRNA molecule and starts the polypeptide chain, and that polypeptide then is anchored to the surface of the ER, and it's pushed through the ER membrane as it's synthesized. The advantage of this is that it doesn't require any additional energy. The energy from pushing it through the ribosome is enough to drive it through the membrane of the ER and into the ER. Now, cells also have what's called smooth ER, and that's endoplasmic reticulum that doesn't have any ribosomes attached to it. In most cells, there's not a lot of it, but in cells where there's a lot of lipid metabolism going on, you find more of it. And this is the location where lipid synthesis occurs. But right now we're talking about the rough ER and the translocation of proteins across the ER membrane. Okay, so we have the two major subunits of the ribosome. The RNA binds, puts the the two subunits come together, and then as the nascent peptide comes through, it docks on the surface of the ER. And you can have multiple ribosomes on the same mRNA molecule, so they can be lined up on the ER, sending multiple polypeptides through at the same time. Again, there's a signal sequence generally on the amino terminus, not always, as we saw earlier. And this interacts with proteins on the ER surface, the translocators, in order to initiate the translocation of the polypeptide through the membrane. After the process starts, then the signal sequence is cleaved off and most likely degraded into amino acids where they can be reinserted into growing polypeptide chains. There's another protein that facilitates this, and this is the signal recognition protein, or SRP. This is a protein, a molecule that is a combination of RNA and protein, and it recognizes, as the name implies, the signal sequence on the nascent peptide and directs it to the translocator. So this is kind of shown here. We've got on the left the ribosome making a nascent peptide, shown in the signal sequence shown in red, and then the rest of the peptide in green. The SRP, or recognition protein, recognizes that red sequence, binds to it, and then brings it over, docks to a receptor on the surface, which then brings the whole complex to the translocator, and the polypeptide can be then pushed through the membrane of the ER. Again, this, unlike transport into the mitochondria, does not require any additional energy other than the energy needed to synthesize the polypeptide. So this is a little more schematic of the ribosomal docking on a translocator, and the polypeptide shown as dotted lines on the right here being pushed through this channel into the ER. Okay, so there are three basic mechanisms for protein translocation. The one on the left that we just talked about, and we find this in bacteria as well as archaea and eukaryotes, and again that involves a signal sequence that's recognized by a protein recognition protein that then binds to a membrane-bound receptor, which then delivers it to the translocator, and it's pushed through as the peptide is being synthesized. The other two mechanisms, one found in eukaryotes and one in bacteria, involve post-translational translocation. In other words, the peptide is made first in the cytosol, and then it's translated or translocated through the membrane, and again in an energy-dependent fashion. So all three of these involve structurally similar translocators, even though they have slightly different mechanisms. Now again, we can have a number of different endpoints for where the protein finds itself. It can go through the membrane of the ER and be inside. It can stop in the membrane. It can be anchored in the membrane with most of the protein in the cytosol. It can be anchored in the membrane with most of it in the lumen of the ER. It can be anchored in either orientation, so the amino terminus can be inside the lumen or outside in the cytosol, or it can be translocated across the membrane multiple times, so it's looped in and out of the lumen of the ER. And here are some examples with signal sequences that direct the protein to different into the membrane of the ER, one with the amino terminus back in the cytosol and one with it into the lumen of the ER. And so there are, again, are a number of different mechanisms that direct the proteins to different conformations using a series of these signal sequences. And this is another example, and I'm not going to go through these in detail, just show you again one more example where both ends of the peptide end up out in the cytosol, and there's a loop of protein in the lumen of the ER. Once the protein goes through the membrane of the ER, it's very quickly glycosylated, and we've talked about these glycoproteins before, that these sugar moieties help stabilize the protein and sometimes help it form the appropriate conformation, but they're also involved in signaling the ER and the Golgi on where the protein needs to go. So they carry out a number of functions. So these end-linked glycosylation shown here, it's the oligosaccharide is anchored to the membrane and synthesized off of a lipid in the membrane, and then it's transferred to the nascent peptide chain as it comes through the transporter. Now, after proteins enter the ER, unless they're going to stay there, they are going to travel through the Golgi to their final destination. Again, there will be proteins that stay in the Golgi as well. These are going to move from one vesicle to another by budding off and fusing to other membranes. And there are a number of molecules involved in directing that. Shown here, these vesicles are coated with different molecules that direct them to different locations in the cell. And you see three examples shown in green, blue, and red for different molecules that coat these vesicles. There are other molecules that facilitate this called snares, and so their job is to interact with the snares on the vesicle, interacts with the snare on the target membrane to bring the membranes closer together so they can fuse. You see this in the next slide. And you know, when you take two soap bubbles and when they come together, sometimes they can fuse to form one bigger one. And that's exactly the same thing that's going on here, only in this case there are proteins to facilitate that fusion as well as other proteins to direct the vesicles to the proper location. So this is a schematic of a pair of snares interacting to bring these two vesicles into close proximity so they can fuse. Now there are a number of other proteins called Rab proteins that facilitate this. docking transport vesicles. And this is a involved slide here showing the steps in the process. Again, we've got a vesicle budding off from the donor compartment at the top. Contains a snare, a rab. It goes down, finds its target compartment. The rab then interacts with a receptor, bringing the vesicle in close. The snares interact, and then the two membranes can fuse together, incorporating the vesicle membrane into the target compartment and releasing the contents of that vesicle into the lumen of the target compartment. Now, there are a number of different RABs that are associated with different types of vesicles, and this is a list of them just to give you an idea that there are many different ones that direct proteins to different locations. I want to talk just a minute about transport from the ER through the Golgi. And again, the function of the Golgi is to direct proteins to their appropriate locations. And one thing you'll notice from this diagram is the arrows between the endoplasmic reticulum and the Golgi are in both directions. So there is recycling of the proteins back to the ER, and we'll see the same thing in going from the Golgi to other subcellular organelles. That the proteins that facilitate that transfer then are generally recycled back to the Golgi so that they can be reutilized. That way you don't have to synthesize new proteins every time. That would be a very energy inefficient process. So this picture shows the formation of a transport vesicle, and you can see again there are these down at the very bottom, these chaperone proteins that hold the what's called the cargo protein, or the protein that's going to be transported somewhere. So if they're soluble, they're sometimes bound to a receptor on the surface. Then they're all congregated into a budding vesicle here. And again, a number of proteins involved in forming that vesicle, or that bud, and budding it off as a vesicle. Then it needs to be delivered to its final destination. And again, that involves these snares that come in pairs. So there's the snare on the receptor membrane and the snare on the donor membrane that again are going to interact, bring these two membranes together so that they can fuse. Now, once again, these vesicles aren't just floating around in the cell hoping to find their final destination. It's very ordered, and they follow these microtubules along a path. So when a vesicle buds off from the ER, it moves along that path to the Golgi, and then when the proteins have finished their job, they then bud off and then move back to the ER again, not floating around hoping to find the ER, but moving along a scaffold. So again, these proteins have to be returned to the ER so they can start the process over and ship more proteins through the Golgi to their final destination. And this shows a little more detail, and one of the things shown here is there's a pH gradient going from the ER through the Golgi, where it becomes more and more acidic as it travels through the process. Okay, this is a schematic of a cross-section of the Golgi, and you can see these stacks of membranes going from the cis face to the trans face. So the start of the Golgi through to the end. And let's look at an example here. In a situation in a cell that's involved in secreting large amounts of molecules, in this case mucus, does it through vesicle fusion, and we'll maybe look at that again in a minute. But once the proteins are translated, go into the ER through the Golgi, they're sequestered in these secretory vesicles, and then given the proper signal, those vesicles fuse to the cell membrane and release their content. So this is a mechanism for releasing a large amount of material in a very short time. This is just showing that the stacks in the Golgi do differ in their content. We can stain them with different histochemical stains, and some of these stains bind to the cis part of the Golgi, some to the trans area, and probably some in the middle. So before we look at the functions going on in the Golgi in a little more detail, I just want to maybe summarize this idea of vesicle fusion and what we need to take away from that. And that's, the idea, again, that once the protein crosses a membrane, it's topologically outside of the cell, or if it's in the membrane, then it's in any membrane. It can be transferred to any membrane in the cell. And these contents then can form vesicles, can butt off the membrane as a vesicle and move to another location, fuse with another membrane, and deliver the contents. And there are a number of proteins that facilitate that, that direct those vesicles to different locations, that facilitate the fusion of the vesicle to the new location, or that actually facilitate the butting off of the vesicle from its original location. And the details of what those proteins aren't so important, but I think it's just good to know the overall process here and the fact that this is a highly structured process. These things aren't just wandering around randomly looking for a home, that these proteins are directed into a specific vesicle. That vesicle follows a directed path to its final destination. It just doesn't float around and hope to find it. So this is a highly organized and structured process so that it can happen quickly and efficiently. Okay, back to the Golgi, and we see at the top proteins coming in from the ER, and then the sorting starts immediately in the cis Golgi. There's glycosylation on many of these proteins, and most, if not almost all, of the proteins that go into the ER and the Golgi are glycosylated. As it moves through the Golgi, these high mannose residues start to be trimmed down. They continue to be trimmed, and then other sugar molecules are added depending on the protein and where it needs to end up. And finally, they are released in the form of vesicles and head out to other locations. Now, there were originally two models for how this worked, and the model shown on the left basically the idea here was that these vesicles budded off from the ER. They then fused to form a stack of the cis Golgi, and then they continued to bud off and fuse to the stacks along the way out into the trans Golgi, and then finally off of the Golgi. The other model was that these vesicles would fuse into a membrane of the cis Golgi, and then it would travel as a whole through the Golgi until it got to the trans side, where it then formed vesicles and budded off. And it turns out neither one of these models is true, that actually the Golgi stacks are all connected to each other. And so proteins move around in the Golgi from the cis side to the trans side just by moving through the membrane. The ER into the Golgi, where they're sorted out and then placed into these vesicles, endosomes, for transport to the lysosome, where there a lot of degradation of different molecules or incoming organisms takes place. And you can see on the right here, there are a number of different types of enzymes that break down different molecules, nucleases, proteases, glycosidases, lipases, so all found in the lysosome, which is also very acidic organelle. So then, as I mentioned, bacteria coming into a phagocytic cell will be engulfed into a phagosome, which then can go and fuse with a lysosome where it can be degraded. Now, some bacteria are able to avoid this process and don't get phagocytized. Other bacteria have developed mechanisms to survive within a lysosome. But the idea here is that if these phagocytic cells run across the bacteria, they engulf it, then send it to the lysosome, where it's degraded and broken down into small molecules. Also, organelles, an example shown at the bottom here, mitochondria have a shelf life, so once they become damaged, they can be engulfed by a lysosome where they're degraded, and this process is called autophagy, or some people call it autophagy. Now there's a receptor that recognizes lysosomal proteins by the carbohydrates on the surface, and these are contain a mannose residue. So this is a signal placed on proteins that are headed to the lysosome to direct them to those lysosomes. So again, the process is shown here. These proteins enter the Golgi, work their way through the folds of the Golgi from the cis to the trans area, where they are collected, and then they can bud off and move into an endosome, which will then transport them to the lysosome. I'm not going to talk about this today because I think Dr. Bartling will be talking about this, but there is traffic going in the other direction from the exterior of the cell into the cell, which is called endocytosis rather than exocytosis, or movement out of the cell. Now there are two pathways to get out of a cell, um, both going through the ER and the Golgi. One pathway goes straight to the cell surface, and the other goes through these secretory vesicles. And in the next picture here, we see the two pathways. At the top is what's called a constitutive secretory pathway, and so these molecules are placed into vesicles and then just head on out to the cell surface where they fuse and release their contents. There's also a regulated secretory pathway where these still form bud off from the Golgi to form vesicles, but they then wait for a signal before they go and fuse with the cellular membrane and release the contents. And this signal can come from outside, which the example shown here, a hormone or neurotransmitter, or they can be internal signals as well. So these are the three best known and major pathways of proteins going through the Golgi. At the top in yellow is movement to the lysosomes, and again this is a signal mediated mechanism. So signals are placed onto the proteins that direct it to the lysosomes. In the middle is the constitutive secretory pathway. So when vesicle buds off from the Golgi, it goes straight to the membrane and fuses with it, releasing its contents. And then the third mechanism is a signal-mediated or regulated vesicular transport to the cell surface. So these, again, bud off of the Golgi, but they will wait around in the cell until they are signaled to go to the cell surface, fuse with that membrane, and release their content. So the default pathway going through the Golgi is to go straight to the cell surface and be secreted. So it requires signals on the protein to direct it to the lysosomes or to a regulated vesicle. Okay, so the process shown here again, after proteins are sorted out in the Golgi, they are formed into vesicles and then directed to their final location. The proteins that facilitate that then are returned to the Golgi to grab some more proteins and send them on through. Now, because these vesicles contain a large amount of material that's very densely populated with these proteins, they are able to release a lot of material very quickly if given the signal. And this is an example shown on the right at the bottom here of a couple of vesicles, one that is just fused with the cell surface and one that's about to, releasing their content. Now, I don't want to go into detail here, but just to let you know that some proteins are processed proteolytically, so cleaved into shorter peptides during the formation of secretory vesicles, and that's the case for a number of, in this case, signaling molecules. Okay, this is an example of a secretory cell, in this case a mast cell, that's waiting on the left, waiting for a signal. You can see all these dark circles are the secretory vesicles, so it's storing a tremendous amount of material. when that cell gets a signal to release that material, it fuses those vesicles with the cell surface and releasing a lot of content in a very short time. And so those of us who have had a reaction to a virus or an allergen have experienced the results. Now, some cells are polarized so that they can release contents in one direction versus the other. And here's a couple of examples. Epithelial cell releasing contents into the outside of the tissue. And then the other one is a nerve cell. And of course, at the end of the axon, where the neurotransmitters are released. So not only can these cells store their contents and release them very quickly, they can direct the contents in one direction or another. Okay, I want to change our target molecules just a little bit, and for the last couple of minutes just talk about sorting of lipids as opposed to proteins. And this diagram shows the initial steps in that. So lipid synthesis occurs on the cytosolic side of the ER membrane. Okay, but if so you keep adding lipids to that side of the lipid bilayer. So obviously we have to flip them over to the other side if at some point, or we'll have more on one side than on the other. And there are a number of enzymes that facilitate that, and I don't want to spend a lot of time on this and go into detail, but just understand that we have the same issues with lipids that we do with proteins, that they need to find their final destination, and all these different membranes, the cell membrane, all these organelle membranes have different lipid compositions to accommodate the function and the proteins that are embedded in those membranes. And so just like proteins, the lipids need to be sorted out and sent to the right location. And this is showing a schematic of that happening. Again, all the lipids are synthesized on the one side of the ER membrane on the cytosol side, so inside the cell, and then they can be flipped over to the other side. And there's proteins that carry this out that can move. In this example, can pull a lipid out of the ER membrane and place it into the mitochondrial membrane. Other enzymes can flip, as we just saw, can flip the lipids from the outer membrane to the inner membrane. Okay, so that's a quick rundown on movement of proteins, a little bit about lipids through the cell. Again, I think it's more important that we focus on the big picture and get an idea of how these things are moving around in the cell, and the fact that they need to move around and get to their final destination, and also the fact that this is a very structured process going on, and these things aren't just floating around randomly looking for their home, but they're highly directed to get to the right place as quickly as possible.